Assalamualaikum and hi, my name is Saida Ifa Hajidah binti Abu Bakar and my matrix number is 67617. My supervisor's name is Professor Dr. Awang Ahmad Salihin. My final year project 2 report title is Isolation and Screening of Lacase Enzyme from Different Types of Fungi. So this is my abstract. As my introduction, bacterial and fungal lacase is proved to productively degrade and decolorize synthetic dyes according to Blanquest et al. 2019. Plant and fungi are the organisms that synthesize lacase enzyme the most. Aspergillus flavus, Aspergillus niger, and Aspergillus oryzae are the fungi which were being used in this research to screen the lacase enzyme. This research have three objectives, which are to grow and isolate different types of fungi, to study the lacase enzyme produced by different fungi selected, and to analyze the quality and activities of lacase enzyme produced. So for the literature review, the first literature review is lacase enzyme. This enzyme is under the blue multicopper oxidase group, and this enzyme takes parts in monomers cross-linking, aromatic cleavage ring compounds, and polymers degradation. This enzyme is commonly synthesized by plants and fungi. Next is for the mechanism lacase enzyme. This enzyme acts as battery which kept the electrons from the oxidation reactions to lower the molecular oxygen. The free radicals can encounter polymerization with the quinone, according to Su et al. 2018. Next, for the lacase methods. Most common lacase detection methods were being used is color detection methods, which involving color changes. The production of the lacase enzyme can be analyzed depends on the color concentration and color circle size produced in the identical time. Next, for the Aspergillus niger, this fungus is white in color at the first rough eyes observation. It turns to black color after several days synthesis. Conidial spore according to Mokobi 2020. When this fungus is grown on the potato dextrose agar PDA at temperature of 25 degrees Celsius, white colonies will appear, but the color is rapidly changes to black color. Next, for the Aspergillus flavus, this fungus have green color at the first rough eyes observation. This fungus have both sexual and asexual production. Next, for the Aspergillus oryzae, this fungus is also recognized as Koji mold, and when this fungus is grown on PDA, white colonies will appear, according to Albashiti et al. 2010. So this research 5 methods, which are PDA solid agar preparation. Approximately 50 gram potato was boiled in 1 liter of water for 30 minutes. Dextrose and agar in 5 gram bowls were added into the effluent. Next, water was added into the 250 milliliter volume. Next, for the fungal isolation, Aspergillus niger, Aspergillus flavus, and Aspergillus oryzae were cultured and isolated from bread, orange fruit, and contaminated rice Oryza sativa L, respectively. Those three fungus were labeled as A for Aspergillus niger, B for Aspergillus flavus, and C for Aspergillus oryzae. Those fungi were cultured for 7 days. Next, for the lacase enzyme production. After 2 weeks, lacase was harvested from the 3 different fungi in the rich media. Next, for the dyeing experiment, a volume of 1 milliliter of crude lacase enzyme extract was mixed with the RBBR dye substrate. The mixture was incubated in water for 15 to 20 minutes. The decolorization of RBBR dye over time was observed and recorded. Last but not least, for the screening of the lacase enzyme, the color changes of the lacase enzyme and RBBR dye from Petri dish A, B, and C were observed and analyzed. For the result and discussion, the first is fungal isolation. Pure fungal isolation was isolated from contaminated bread, contaminated orange, and contaminated rice on Rhizosativa L. The pure culture were obtained from the PDA culture after 14 days. The figure shows of the culture plate A, which is the Aspergillus niger. Next, on the plate A, the black color of mold was first ground on the first 7 days. Another 7 days, the gray color of Aspergillus niger was observed. Next, for the picture shown is the culture plate of plate B, which is Aspergillus flavus, after 40 days. The brown color mold was first ground and green mold of the Aspergillus flavus were appeared after 14 days. The color of the colony should be green, but if the first deep brown color is shown because of the sclerotia pile of fungi. Next, for the plate C, which is Aspergillus oryzae. On the plate C, the white color mold is observed after 14 days, as you can see in the picture. Next, for the lacase enzyme production, each of the different fungi isolated was further cultured into liquid media on another petri dish for 7 days. Lacase enzyme was assumed to produce in the liquid media. Next, for the screening of the lacase enzyme, a volume of 1 milliliter of crude lacase enzyme was isolated into another petri dish and was mixed with RBBR dye. Plate A, B, and C were incubated on warm water for 20 minutes. Unfortunately, there are no decolorization occur in either of the one of the 3 dishes. The time count was extended into 2, 4, and 6 hours to observe whether there is any reactions happen. So this is the observation of lacase enzyme with the RBBR dye after 6 hours. As you can see, there are no decolorization happen on all of the 3 plates. Lacase enzymes are considered non-active in those 3 petri dishes. It is required to enhance the catalytic activity of lacase enzyme in order to increase the abilities and activities of lacase enzyme to decolorize the reactive dyes, according to Vantamuri and Kaliwal 2016. One of the probability may due to the lack of instruments and some materials which are required to screen the lacase enzyme properly. To improve the studies, some appropriate methods can be done, which are for lacase enzyme purification, ACTA primer purifier is needed, spectrophotometer instrument is needed to analyze lacase enzyme activity. Lacase enzyme activity and quality can be observed and analyzed depends on ABTS at 420 nanometer. In brief, the objectives are not totally accomplished due to a few shortages in terms of materials and equipment which required. There might be contamination in the culture that cause inactive or absence of the lacase enzyme. The 3 different fungi is successfully grown on the PDA solid agar even though requiring long time than expected. So these are my references. That's all for my presentation. Thank you.